Hello, everyone. Today we are going to talk about zebrafish and precision medicine with application in Parkinson disease. So I asked ChatGPT to create a picture for the title of the talk today, and it came with such a nice picture. And it's about TRANSFAC and about networks and searching for master regulators. And actually, it's about zebrafish and zebrafish in the head. So which means the application of zebrafish in Parkinson disease. All right, so let's proceed. And actually, why zebrafish? I put this very nice picture from this company. I grabbed it from the internet. As you can see, the zebrafish brain is very colorful. It expresses many different proteins. And why should we study that? Because it's a very good model organism. And actually, it's often used especially for studying neurodegenerative diseases, because the brain of zebrafish is, although it's very, very far away evolutionarily from human. Let me see if I have everything set up right. But still, it has the areas in the brain which can be used to study different diseases related to neurodegenerative diseases like Alzheimer's disease, because the areas of the brain of the human brain are also represented in the brain of zebrafish, and especially for Parkinson disease, there are special parts of the brain which are affected by this disease, and the neurons are dying in that part of the brain, and this mechanism is studied in zebrafish. And why, in general, we are talking now about such model organisms? Because just recently, in our new release of TRANSFAC, we came up with a new tool. Well, it's the same tool as before. It's called Match Suite to search for transcription factor binding sites, and we made a nice effort just last a couple of years, and we input there possibility to analyze promoters of some model organisms, and among them the zebrafish, and I will demonstrate today how to do it for the genome of zebrafish. And as I said, in general, why do we need model organisms, especially for such diseases like neurodegenerative diseases? It's very difficult to study them in human. The only way really to get into the tissue often is related to basically post-mortem tissues, when it's already too late to understand what's going on. Especially nowadays, when we want to have like a precision medicine, which means by measuring a lot of parameters of the living body of the living human, we can predict what kind of drugs it can be used. But if you can get only tissues from the brain, it becomes very difficult to study such problems and to apply such tools for a human directly. You have to do some model organisms or model systems like cell lines. And in case of zebrafish, which is represented here, you see there are areas of brain which are very similar in terms of producing those types of neurons. And for the Parkinson disease, especially, we are talking about neurons which are actually producing dopamine, and they are especially sensitive in case of Parkinson. And that's why the zebrafish is very useful to study such kind of disease. And this is another picture which shows also similarity between the brain of human and zebrafish. And okay, coming to Parkinson disease, you know that's a very important disease. Many people suffer from it. And it actually relates to such symptoms like tremor and many other bad symptoms, which are actually accelerating with age, or this can be, people can be affected even quite early in their life. And it's still very, important to understand. So there are no very effective medications so far to really cure this disease. And one of the main mechanisms which was which found to be related to the Parkinson disease, is a mechanism of neuron dying because of some amyloid fibrils, which is supposed to be accumulated in those neurons. And for whatsoever mechanism, those amyloid fibrils are becoming, first of all, they accumulated in those neurons, and also they actually are able to trigger dying of neurons of nearby, so of the tissues nearby, so they kind of infecting other neurons. So, and this kind of mechanism was in our focus when I was in one of the projects recently related to study Parkinson disease, where partners of that project, they found out that they can actually generate those fibrils in vitro, and they can, in fact, by those fibrils, they're called fibrils of alpha-synucleins, of special type of them, and then they can actually in vitro on cell lines. We were able to see that those actually neurons, which are infected by those fibrils, they start dying. And this was a very big project with many partners from the whole Europe. And we did a lot of omics data on those cells, and we used the human cell lines and also iPS cells generated from Parkinson disease people. And we found out, I want to show this network. First, I will show the general picture, and then I will dive a little bit inside this network to show the details. Actually, as I said, we generated a lot of omics data, especially there was a transcriptomics, before treating by these fibrils and after. So actually, after treatment, we also observed that those neurons were dying. So definitely those fibrils were leading to neuron death. And then in addition to transcriptomics, there was the proteomics made, and also interactomics. It's a special kind of proteomics when we actually see which proteins are interacting to each other. And also there was done another type of proteomics, especially for mitochondria, because that project was actually focused on mitochondrial influence on the dying neurons. And if you look at this network more in detail, I have to zoom it like this. This network was produced by us, by geneXplain. And here at the bottom of this network, you see those genes, which are differentially expressed upon treatment of the neurons by those fibrils. And the blue guys here are the transcription factors, and we'll talk about them today much more. And on top of those transcription factors, we see this big network of signal transduction, which leads to the activation of those transcription factors. And the whole story was to find so-called master regulators. So those proteins in the cells, in the neurons, which are increasing their either expression, or they are shown to be highly upregulated in the proteomics data. And if they are at the top of the pyramid, which means the activation or inhibition of those proteins can lead through the signaling cascade down to activation or inhibition of that set of transcription factors, which in turn will actually influence the expression of those genes. So these are obvious targets for the drug therapy. And among, one of the top master regulators, we actually see this FIN kinase, and it came up in my analysis of master regulators very frequently, and I showed it to my colleagues, but they said to me, no, it's very old and boring kinase. We will not use it as drug targets. We'll rather use another very specific ones, and that makes sense. But still, you see, it's really playing a role, central role in this network. And I was really happy to see that recently, and I'm going to show today. I found the gene expression data, which was recently produced. Let me open this gene expression omnibus entry. It's actually November 2024, so a very recent paper. or the paper is actually not yet published, the data is already released. It's actually a very good chance to analyze this data before it's even published. And here, what these authors of this paper did, they actually created a model, a zebrafish model, where this fin kinase basically it was introduced in this model. So it was a knock-in model. So where this fin kinase specifically phosphorylated, so getting active, is introduced in this organism and is getting kind of constitutively active upon a certain signal. And then they obviously can see, well, it was not obvious, but they have seen that actually those neurons, especially those which are producing, which are responsible, the neurons which are responsible for the Parkinson disease, they actually dying. They start dying. And they also found out that the intermediate mechanism of that is related to actually inflammatory and immune response. So I was very happy to see that, and that's why today we are going to analyze this data using our tools in TRANSFAC. But before going to that, let me first show a little bit of the theory, how we are going to analyze those kind of data. So first, we'll take promoters of the upregulated genes, and we'll analyze their, the transcription factor binding sites, enrichment in those promoters. Sorry. And especially, we'll do it in zebrafish genome, which becomes now easily done in using our MatchSuite tool. And after that, we'll run an algorithm to search for master regulators. And I will show you a little bit, and I will focus on today on that algorithm mainly. And this algorithm allows to find master regulators, and from master regulators, we actually can predict which drugs can be used. And this part is actually done already on human data, on human network. Because actually the signal transduction network is well studied on human, mouse, rat, on those model organisms, and not well studied on some small organisms like zebrafish. And that's why here we get the transition from the zebrafish to human and to the precision medicine, which means we'll be able actually not only predict the general drugs which can be applied to for the treating Parkinson disease, but we'll be trying to make a precision medicine, which means in a sense a personalized medicine to actually see priority of which drugs can be applied to which of the patients. And that's all together, starting from zebrafish to human and to drug predictions. All right, how we analyze these kind of networks, as I mentioned before, so we start actually, sorry, it's going automatically. We start from a list of transcription factors which we predict by analyzing promoters, and we did it many times. I showed it before. But then when we have in these transcription factors in our hands, the question is, which are the master regulators which can lead in a single transduction network to activation of those transcription factors altogether. And we have these upstream analysis algorithms which are running in this graph of signal transduction network, which we collected in TransPass database for many years. And using this scoring function, we find which of the master regulators potentially can reach maximum number of these transcription factors through that signal transduction network. And this scoring gives us a kind of a scoring and ranking of those master regulators which we want to find. But this only half of the story, because the master regulators can be found quite many, because this network goes quite far away. So at the moment, we have collected more than 1 million reactions from different papers, which which are building this huge graph, huge network. So that's why we have give additional priority to some of the master regulators, and which of them, for that we found actually, we formulated a so-called walking pathway methodology, where we found out actually that these signal transduction networks, they are very flexible. So in normal cells, in normal neurons, the neurons receive signaling, and then the signaling leads to activation of certain transcription factors which regulate gene expression in normal situations. In disease, rather, this signal transduction network is often reconstructed or rewired. So, and we found out that very often new master regulators pop up in disease condition, which in a very important feature of those master regulators is that they actually connected to the genes which are regulated by themselves through the positive feedback loops. So, which means that activation of such master regulators lead to the activation of the genes which encode those master regulators which leads it to the never-ending loop. So, and very often this healthy status of the cells or the organisms is related to the disease states, just in this numerous number of the feedback loops, especially positive feedback loops, which stabilize this disease state of the system. So that gave us additional possibility to select those master regulators, which are not only can reach signal from those master regulators can reach our set of upregulated genes, but it also characterized by multiple positive feedback loops. All right. And that's why we call them walking pathways. So, which means basically the signal transduction network is more or less the same, but very often the bottom part of that pathway is corrupted or changed. So this set of terminal kinases is starting to activate a new set of transcription factors, which are now activating a new set of disease genes. So using this concept, we basically realized how to analyze such data. All right, so now let's go online and see how this can be done online. All right, so as I said, we have this entry in gene expression omnibus. And as I said, what they did, they had this Zebrafish, which model where this FIN kinase in active form was introduced in the model organism. So they're expressed in those animals. And then there's of course, there's a wild type control and also other types of controls. So what we are going to do, we're going to compare these genes which are expressed in the model organisms and compare them to the expression of those genes in the control. And to do that, we have to download data from gene expression omnibus. So they showed here, they actually measured it in different conditions, like after 3 days, in the model organism and control, and also 5 days. So let's take the 5 days, so the very end. So after 5 days, definitely a lot of changes could happen. Let's take this 5 days data and analyze it. So basically what we did, we simply took this entry. Let me see which one. So I think this one. So this is basically we just clicked on it here and downloaded it to our computer. And then let's go to geneXplain and upload this data. So first, let me go to the file which was downloaded. So let's first unzip it. So now you have here this file. So we can actually simply just to have a look, what is it, what is the structure of that file. Let's put it in Excel, and just to for you to see what kind of data we have here. So in the first column, we see here the genes, actually ensemble ID of the zebrafish genes. And here we have the replicas of the model organism and 4 replicas of the control. And actually, it's also already calculated p-values and a fold change between them, but we'll do it again in our system just to see how it works and to do the whole thing consistently. So first thing we have to do is to log in into system, and you've seen it before, how to log in. And so I have here this folder for the project today. So this coffee break from zebrafish to precision medicine. And what we can do, we can upload here. So let's create a folder here. Folder, so meeting. All right, and so we will actually load this data into this meeting folder. You can load it directly from your computer, or maybe even directly from FTP side if this file is very big, but in this case, it's not very big, it's very small. It's actually recognized that this is a data from the zebrafish. So, and it's already found out that it's ensemble IDs. So it's the only thing you have to do is just click start. And now you have this in your system. All right, so the next is to find differentially expressed genes, right? And to do that, we have actually a number of tools here in the geneXplain platform. And we now go to statistical analysis tool. And here we start Limma. Let me put this aside. And what we are going to do, we will take this data here. We'll actually define that it's not the logarithm, it's actually an unnormalized counts, as far as I know. And now we can, say this is a model, and these are the model data for replicas, and this is a control, and these are the control data. All right, and that's it. So then we can simply run it. So now it's actually using the R script and taking the Limma package from standard Limma package from it's not something what we wrote by ourselves. These are very standard packages from bioconductor. They are all integrated into the system, and as you may know, in this geneXplain platform, we integrated a lot of different tools. Some of these tools are actually written by ourselves. They are written in different languages, in Java, in Python, in R. There are more than, I think, several tens of this tool, maybe even more than 100. And they are in statistical analysis, in functional analysis, in side data, in promoter analysis, which we are going to do today, and also tools for network analysis, so which we have also here. All right, so each of that tool can be used, and some of the tools are actually used from the Galaxy partition of that geneXplain platform. So we have a here interface to incorporate any Galaxy tool. So, and we have a number of Galaxy tool for RNA-seq analysis like TopCat and others, or from peak calling in case of ATAC-seq and ChIP-seq data and so forth. So that's all you probably know, because you are probably users of our geneXplain platform. If not yet, just go to our website and register for geneXplain platform. So this calculation, I'm talking because I'm waiting for this calculation to finish, but I see it's still taking time. That's why I will go to pre-calculated results, which are already here. And what we are going to see now is the results of this, Limma analysis, which is in this file. So to open the file, you just double click on it. And now you see all the 25,000 genes in Zebrafish, and you can sort them by log-fold change, for instance. So which means at the top of the list, you see the very highly upregulated genes, and you see also p-value and adjusted p-value and all kind of characteristics. So basically we see here upregulated genes and downregulated genes. And we can also do different kinds of filtering to define upregulating and downregulating genes for our further study. But what I want to do now first, I want to ask a general question. By the way, this is a results of my run, and as you can see, it opens automatically, and actually results are the same. You see up and down regulated genes. So let's close this table. What I want to do is I want to actually understand in general terms what kind of changes in functional terms happens in this zebrafish in the model organism upon activation of Fyn kinase. All right. So to do that, we go first to the analysis tab, and we will open the functional classification category, and we'll do actually enrichment analysis. And this is nothing, especially different from what you may usually use. It's a so-called gene set enrichment analysis algorithm. Here it's simply very easy to start it. So you take your set of genes. So like we have just created here, already computed log-fold change. You drag and drop it here. And it already identified it's a zebrafish. And as I said, in this release of TRANSFAC, we added number of model organisms, and you can do the gene ontology or other types of categorical classification and gene set analysis for different model organisms. So I actually just have a look at which organisms we have introduced. You see this fruit fly, so Drosophila, of course, human, mouse, a rat, nematode, all kind of yeast, zebrafish. So that's the set of organisms we are now can work and do all, many kind of analysis. All right, so what I'm going to do today is actually to especially look for gene ontology terms, biological processes. And we'll try to find such biological processes which are enriched among the upregulated genes. And okay, so basically these are the parameters of the search, and I will run it. So again, it may take some time. So let me just open for you the results, which I did already before. And this is a type of results you get. So actually, if you are familiar with gene set enrichment analysis, you may also see here how does it look like. So for instance, if you see here big categories, for instance, the whole cellular biosynthetic processes, or like primary metabolic processes, so a very big category. But of course, if you roll down and you see more specific ones, and that one which is really attracts my attention is immune system processes. And as you remember, so the whole what authors of that paper also saying that after activation of Fyn kinase, these immune cell processes they activated, and they lead to death of those cells. So if you look at how does it look like in terms of gene expression, gene set enrichment analysis, you know how this method works. Is actually taking genes from a particular ontology term, and then you sort those genes by log-fold change. Here at the left corner, you have upregulated genes, and in the right corner, you have downregulated genes. And then this is actually the blue line shows the logfold change. And you see those which are around the zero, it's non-change genes. But those which are here in the left part, they are upregulated genes. And then red line shows it goes each time up as soon you get here the gene from immune system process. And then as you see, you observe if this curve goes very high, then it estimates also the p-value of that and tells that it's a trend of enrichment of that genes belong to that category at the left part of your curve. Right. So and actually, I'm often what I do it sorted by rank at max, which means I'm interested to those cases when the maximum peak, for instance, like in this case, like in this case, you see the maximum peak is at the very left corner. So and which means the genes which are belonging to that category, they are in the very beginning of the list. So they have very high logfold change. And right, so and then you see here immediately, some categories like complement activation, or again, many different categories. What I did, I actually sorted and filtered that by p-value, by FDR, and by this rank and max. And in fact, what I did, I collected here those categories which have a maximum impact, so maximally enriched in these upregulated genes. And what you see here around immune response, a lot of subcategories of immune response, defense response, again immune response, and many of those categories are actually getting enriched here. Why I'm talking here about this enrichment so in details, because what we are going to do now, we are going to apply this knowledge that these genes are enriched and the process of immune activation is activated and working upon this fin activation. We will use it in our match suite tool. Let me show how we are going to do that. But first, what we are going to do is we want to select a set of genes which are especially interesting for us. As I said, we sorted by max and rank. And what we do also, let me find a gene set which has a very many components. So this is, for instance, response to external stimulus is pretty general term. Maybe it's not so interesting. So let me go a little further. I see here a term where it is for a response to bacterium. So I'm looking for those which have a very big number of hits. So it means genes belonging to this category. So let me basically filter it by immune response. Let me put it here, title immune response. Yeah. So you can filter this list, or you can sort it by different parameters, for instance, by logfold change and by p-values. So here you see all these immune response categories which are enriched in our analysis. For instance, this category, and now I want to set, define this set of genes. So what I do simply, I mark this line, and I export this set hits of that immune response genes, and bingo, I have them in my repository. So these 440 genes, which are belong to the immune response, and you see here the logfold changes for these particular genes. All right. Why do I need that? Because I want to analyze promoters of those genes. I want to analyze why those genes are actually, upregulated. What is controlling the expression? All right, so that's you can do here in our tools. For that, we return back to our start page, and we will start our match suite tool. which will be able now to analyze such sets of genes. So what you have to do, you have to upload this set of genes in this tool, and it will analyze promoters of these zebrafish genes. All right. So for that, we go to this start analysis. We select here the zebrafish, and we will use here a set of genes which we have already copied. So here I already copied this set of genes. Let me see whether they are them. Immune response. I selected also upregulated genes of them. So in total, we have 98 genes which belong to this immune response, and they are upregulated. So their log-fold change is above 0. And okay, we'll take this set of genes, and we'll proceed further. So we want to analyze promoters of those genes to understand transcription factor binding site enrichment in the promoters of those genes. So first we have to define the size of the promoter we are going to analyze. So we are doing it from 100, from 1000 to 100 from start of transcription. And here is a point where we have to define which particular process in the cell we are interested in. So we are interested in processes which are related to immune response, to defense to the external organisms. So those particular categories which we found enriched in our experiment. So that's why what we do here, we select those terms like immune response terms. Like, let's just select all of them. So in this system, we selected those which are have been connected to at least one transcription factor, which is expressed in those which is linked to this kind of function. So we also can add here, like defense and defense response, for instance, and so several other those categories we found interesting, and we want to actually analyze promoters of these genes in terms of enrichment of transcription factors which are acting in that particular function. So which are for acting in the immune response. So we go further next. Okay, so we have the similar name already. So I have to give a different name here. You can do further selection, but the major thing, you simply go next and you launch the analysis. So it will take some time to sort it out. First, it will define promoters of those genes from the zebrafish genome. It will actually take transcription factors which are connected to the selected gene ontology terms. So which means it will make a focus on those transcription factors which play a role in those immune response mechanisms. And then it will start searching for the binding sites for transcription factors in the promoters of those genes. And also it will do many other things. But of course, I will not bother you with all these results, all waiting for the, until this program is finished. I will open to you the final results of that search. It's here. All right. So what you will see, what we are seeing here is a list of transcription factors which are whose sites are enriched in promoters of that upregulated genes. So here at the right, we have this a set of 98 upregulated genes. And here at the left, we have a list of transcription factors whose sites are enriched in the promoters of those genes. We actually found those sites using these motifs from TRANSFAC. And we found out of actually around 8,000 motifs, which we have in TRANSFAC. We found here 13 motifs which are enriched in promoters of these genes. And already on the motif side, you see, for instance, this top motif, which is RF8, so interferon response factor. which is very significantly motifs. So sites for these motifs are significantly enriched in promoter of those genes. And this is the first part. But here what we see is actually a list of transcription factors which are connected to those motifs, like this motif of RF8 was built on the sites for RF8, but actually this motif is connected to the set of transcription factors which are of IRF family. And which of these factors are most important for the process of immune regulation or immune response are actually shown here in this last column. If we score, for instance, sorted by the score, we see, for instance, this factor, this transcription factor, although the motive for this factor was not really, well, it was enriched in the promoters, but not the top enriched. But what is interesting, this factor is connected to immune response genes very tightly. So the score of that connection is very high. So, and basically when we sort these transcription factors, but by this kind of balance between the site enrichment and the belonging of this factor to that particularly selected geo categories, we see at the top those transcription factors which are most important for our analysis. And actually, one important point here to see how these transcription factors can work together. But first, let's have a look at the report. So this tool is also generated, an easy report which you can, print or save on your computer. So, and you can see here all the intermediate steps of the analysis. You can see this list of transcription factors. Also, you see the maps of those sites on each of the promoter. And here also you can see important part, which is so-called composite elements. So this program is also built a frequent combination of those transcription factor binding sites found in that promoters. It also shows how well this frequency of this combination discriminate these promoters from the other background promoters. And now what you can see here, it actually found out that such motifs like IRF8 or REL-A, which is a motif for NF-kappa B transcription factor, as well as a MIP motifs and some other motifs are actually frequently found in combination. So we can indeed can have a look at it more in details when we look at the particular genes. For instance, if you look at this and these genes at the top, let's have a look at this transcriptional at the immune response regulator A gene. Here in this table, you see which of the motifs and transcription factors sites for these factors were found in the promoter of that gene. And then, for instance, you also see that the factors for the NF-kappa B sites, these REL-A sites were also found here. And basically, you see that in the schema. So you see this gene here, the first exon of that gene, it goes in the minus strand. And here in the front of that gene, there's found binding sites for transcription factors. And you see this REL-A binding site, for instance. You see it, in more details of that site. So here's the consensus, so the logo plot of that matrix, and here is a score of that site and other information. So you see also here 2 MIP sites around, so other sites for other transcription factors found in the promoter of that gene. So it's you see it's not only finding all potential binding sites, this kind of information you also have, but it actually selects those transcription factors which are related to the process which is in focus, in this case the immune response, and finding this binding sites and composition of them in the promoters of those genes. So that's we did it in Zebrafish. And if you have now any questions concerning the zebrafish story, it's not yet done because, as I said, from zebrafish, we are going to go now to human. And then we'll see what are the relationship between the results we found in zebrafish and what we are going to find in a human. But if you have any questions so far, just put them in Q&A, and I will try to answer them. But in the meantime, let's move to the human. So what is about human? Actually, now we are going back to our platform. Let me see, where do we have it? Here. And if you did, you see, we found these upregulated genes upon immune response, belonging to immune response, and they are upregulated upon activation of Fyn kinase. All right. So there are 98 genes in zebrafish, and let's actually find which are the homologous genes we have in human for the same zebrafish genes. So what we do then, we have here among the data manipulation, we have special tools which can do that. So what we do here is convert table via homology. All right, so what we do is we take this set of genes, put them here, and this will be then converted to the human genes. Let me see, right, okay, so it detects this is zebrafish, this is an ensemble, and we want to convert it into human genes. So we simply, put here the name for the human genes. Okay, let me put 2 so that we not override them, and simply we run it. So it will actually let me open the results of that conversion. So it found not all of those genes in the human genome. It's only 60 genes. Well, some of the, actually, these are 2 genomes which are quite diverse from each other. So, we also talk about gene families. Sometimes you have paralogs in one genome, and only one of the paralogs have the ortholog in another genome. So such a direct comparison of such a distance genome, you lose quite a number of genes. But still, we have quite a good set of genes here. And what we see here, they are all, belonging to human genome. So now what we can do, we can actually ask a question. Can we find master regulators in that, for those genes in human genome? And to do that, we have this tool, which I already showed before, which is called Genome Enhancer. And what we are going to do, we are going to take this set of genes and run it in Genome Enhancer fully for a human genome, and ask a question: can we find master regulators of those genes, which are activated in zebrafish upon Fyn activation, and whether we find any similarity between human and zebrafish in such comparison. So, and what we do now, we go here to this folder, to this project. We don't need to upload here any set of genes because we have it in the system already. So we click next, and basically I have already done many things here. So what I did, actually, I took this gene set, which we generated, so meaning these 60 upregulated genes, which are human genes, which are upregulated in zebrafish. So, and I simply drag and drop them here. They are here, and then I click next. And actually what I did, I took this log fold change, zebrafish log fold change, and put it in here. And then I click next, and here I selected Parkinson disease, and also I selected all kind of neurodegenerative diseases. Why? Because I want to be a little bit broader, not just finding those drugs which belong to Parkinson disease, but actually looking a bit more broader for all kind of neurodegenerative disease. Maybe I'll find some drugs from another diseases which may be also applicable for the to cure the Parkinson disease. All right, and then I simply select this category and I clicked run. So, and of course, it will run for quite some time. It takes a couple of hours, maybe 3-4 hours to finish such a run. Well, with a small set, it will even faster. And that's the results which I obtained. And as you can see, the system found Fin gene as a top master regulator. Although, as you remember, the experiment was done in Zebrafish. We just took these upregulated genes in a special category for immune response, and then converted to the human genes and run this analysis in the human gene, and we found the same effect, which shows actually very close similarity between these systems, although they are very distant in evolutional distance. But these very basic mechanisms, especially related to immunity and innate immunity, it's very, very common. So, and I will not go through all these, results which we found here. Just to want to show, first of all, these results of, again, composite element analysis now in the promoters of human genes, the same genes, but in human genome. And what you can see here, what is actually interesting, you see, for instance, real A. So other factors which are not found in zebrafish, but some of the factors, some of the key factors, especially real A, it's a transcription factor from NF-kappa B family, which is definitely involved in immune response, also found here in the promoters of the human genes. And then what we see here is a full diagram how this mechanism look like in the human, regulatory system, where we have here at the bottom, we have these upregulated genes. We found these transcription factors, which are, whose combinations are found in the enriched in the promoters of those genes. And upstream of those transcription factors, we see these kinase cascades, and at the top of it, we have a number of kinases, and among them, the Fin kinase as one of the most important master regulator in this whole story. All right, so at least we see the similarity between this zebrafish and the human system. But now, what about precision medicine? So what is a precision medicine, actually? Again, at the very beginning, I said what we want to do is to understand the mechanism of disease from the molecular point of view, from the precision methods like measuring expression of many genes in genome, and then based on those results of measuring with a high precision for each individual patient, or maybe for cohorts of patients, to predict what would be the best therapy for those people. And okay, so then in the practical terms, what we are going to do, we can actually now define this core mechanism of that disease. Maybe, of course, it doesn't cover all aspects of the disease. So it only immune response, and we actually see that this Fin kinase is one of the key regulators of immune response related to the death of those neurons. So assuming that this is one of the core mechanisms, we can basically apply this core mechanism to just patient data and see which patient, in which patient we, of the, of a particular disease, in this case, Parkinson disease, we see which master regulators are actually activated. So, and depending on which master regulators are activated in particular patient, we can prioritize different drugs for these different patients. Okay, how to do that? For that, we go back to the platform, and then we will look at this in this network more in details. And we'll actually simply take these genes as a set of genes. So what we are going to do, we take data from another gene expression omnibus entry, where we have actually Parkinson disease patients. So I found this entry in gene expression omnibus. This is actually not, a paper from 2019, not yet published though, where there were 4 patients, or actually 5 patients of Parkinson disease. And what these authors did, they actually, of course, they cannot, these are living patients. They cannot get material from their brain, but what they can do, they can take blood from these people. And in the blood, they take lymphocytes. Why? Because actually, lymphocytes in the patient blood can reflect processes in the many organs of the person, and also in the brain, especially if there's a immune processes going on in the brain. Very often, what happens in the brain, there is actually immune-like cells which are acting in the brain, so-called glia cells, which are involved in immune response, and they are very similar, and they actually also related to the lymphocytes in the blood of those patients. So by analyzing the blood, the lymphocytes from the blood, we can actually try to see like a mirror what's going on in the brain. So what they did, they have these 5 patients from this blood from the patients, and they have also 5 healthy controls, and that's just gene expression data. So I have loaded them in the platform. All right. So what we want to do now, we want to look at these patients of Parkinson disease. Actually, I loaded them in a separate project. They're here. And so these are the basically this data from those patients, where there's just 4 patients, this PD1, PD2, PD3, so 4 patients, and these accounts from immune cells of those patients, and there are also controls, and then using this tool here in the method section, which is called statistical analysis, and calculating the fold change, which allows actually to calculate the fold change for each individual patient. So you take this the whole set of data here, and you define which are your actually your patients and which are your control. And then you are saying, I want to average all controls and take each patient separately. So as a result, it actually return such a table where you have, for each patient, you have log fold change compared to the control. This is one thing. So actually, I did it for the whole set of data, which is here, right? So like 20,000 genes. But then what I did, I selected only those genes of them, namely these 27 genes, which are found as master regulators in this zebrafish experiment. So again, we're returning to the zebrafish results. So we got this, zebrafish upregulated genes, and then we run them to find master regulators. And here I show again these master regulators to you. Just a moment. Here are the master regulators. Basically, all master regulators which were found there. And now what I did, I actually put here the all expression there from the patients, from these 4 patients, from the blood of the Parkinson disease patients. So now, for each of these, for this is a master regulator, you see this 4 stripe, 1, 2, 3, 4. And this color of these stripes related to the expression of that protein that master regulator in that particular patient. And you see, for instance, this master regulator, so this chemokine CR receptor 4, is actually upregulated in all 4 patients. And also the FIN is actually upregulated in all 4 patients, although in the patient number 2, it's upregulated more. But for instance, this one is in the last 2 patients is actually downregulated. So you see these different patients, they shows a different, slightly different priority of those master regulators. And this information can be used by our system to give a priority to the drugs. So to make the whole story short, I just show you the final result of these 4 patients with a focus again. So I made a focus for these four patients just on these master regulators found from the Zebrafish experiment. And so this is for the first patient, the final report. So it found that this patient has these 2 master regulators, is most upregulated, and the recommendations for these patients, the priority of the drugs for that patient depend on the priority of master regulators. And you see these are the drugs, the Parkinson disease, either already approved or those drugs which are on a clinical trial, sorted by this priority of the master regulator expression in this patient. And for the second patient, we can also see these drugs. You see the second patient actually very similar to the first one. And the sorting of the drug is very similar. So as you see here, maybe a little slightly different. So this is the first patient. So the second drug is in the priority is this one, but in this patient, it's another drug. Although it's number 3, you know, it's a little bit of change of priority. So if the doctor would be deciding which drug to use, it may use this information for the decision. Although the first priority is in these both cases is this drug. But I don't remember which patient was really different. I think that the last one. Right, you see even the master regulators were found slightly different in that patient. And if you see the drugs, well, still the sorting of the drugs is similar. So drug recommendation is quite similar, although the details of the mechanism could be somewhat different in different patients. This reflects actually 2 things. One thing is since this mechanism, although being quite somewhat different, the core of the mechanism is very similar. That's why drugs at the end are quite the same. But also that reflects the limitation of drugs so far which are on the market. So which means there are drugs which are actually targeting particular targets, and that's it. There are no drugs which targeting maybe other targets which might be important for us, but simply there is no drug on the market. So that's why, you know, what could be done is actually looking at the secondary list of drugs which are not actually yet, approved for Parkinson disease, or not yet on clinical trials, but they are from another disease, so-called repurposing drugs, and they are quite different for different patients, and that's why they can be used for prioritization. All right, so as you can see, we started just from a model organism, Zebrafish, and we show that there is quite a high similarity between the mechanism of a neuron death related to Parkinson in these 2, more in the human and Zebrafish, and then we use this information to do some kind of personalized medicine. So at least giving a priority to drug therapy, which can be useful and can be taken by the doctors. All right, so that's it for today from point of view of giving my information. Again, if you have any questions, I can answer them. All right, so if there are no questions, just send us questions then later. And if you are listening us in the YouTube, also put like, and that's very helpful for us. Thank you for your attention and see you next time.